Okay, hi everyone. This is our lecture on chapter 5, which is membrane structure and function. Why is it not switching slides? One second, for some reason it's not. There we go. Okay, in terms of, so you know how the chapters always have a little introduction. So this introduction is actually about something called capsaicin, which is actually a substance that's in hot peppers. And so that's the substance or the chemical that basically makes your mouth feel like it's on fire. And capsaicin, this molecule, binds to a protein on the plasma membrane, right, of the pain receptors in your mouth, and that's what causes that feeling. When that capsaicin binds to its receptor sitting on the plasma membrane, right, of those cells, the receptors open channels and calcium ions can move through them into the cell. And basically, the movement of those calcium ions causes the pain receptor to send a signal to the brain, and then the brain interprets that as pain. So there's a couple themes here. We're talking about membranes, right, this chapter. And within that plasma membrane, there are a lot of different types of membrane proteins that sit within the membrane. And one of those types of proteins are receptors, right? So any type of drug that you'll take, a pharmaceutical drug or otherwise, is going to bind to some sort of receptor to have its effect. And obviously, if we're talking about, like psychoactive drugs, they're going to bind to receptors on neurons, right, on cells in the brain, okay? But in this case, we're talking about this chemical capsaicin, which will bind to pain receptors in the mouth, and then essentially activate them, and that's what's causing that sensation of pain. All right, so we'll talk about some membrane proteins. We're not going to talk about in depth about receptors, right? We'll just sort of mention them and say they are a type of membrane protein. That's something that you talk about more and more, again, as you move on and get into sort of more detailed types of classes, like a cell biology class or what have you, or neurobiology. One thing that we will do towards the end of the course is to do a presentation, and the presentation is actually involving these concepts, right? A drug binding to its receptor, and then how does it have its effect? And so we'll probably talk about that in the next week or so, because it does sort of tie in with this chapter. All right, so anyway, moving on. Okay, so in terms of outline, we'll talk about the plasma membrane and its structure, and then we'll talk about transport across the membrane. So we'll talk about passive transport, active transport, and then we'll talk about some modifications of cell surfaces. Okay. So in terms of that first part, structure and function. So what is the plasma membrane composed of? Okay. Well, in terms of basic function, right? Well, one, all cells have a plasma membrane, right? And they're made up of predominantly of a phospholipid bilayer, but then there are other components as well that we're going to talk about. In terms of just its general overall function, it's going to separate the cytoplasm, right? Internal cytoplasm of the cell and all of the organelles that we talked about in the last chapter from the external environment, right? It's only going to allow certain molecules to come in and certain ones won't be able to. So in terms of phospholipid bilayer, right, I'll show you a picture, and we kind of already mentioned this when we talked about lipids. So the external surface is lined with those hydrophilic polar heads, right? And then basically I'm both facing the outside of the cell and also facing towards the inside of the cell. But then in the middle of that bilayer is where you have the tails, right? And so that's what this says here. Those non-polar hydrophobic fatty acid tails are sandwiched in between, right? And so again, we'll see a picture and I'll point out all these things in a second. And so like I said, it's not only made up of these phospholipids. That is a major component, but there are other components as well. And so again, we just talked about the lipid component, which is that phospholipid bilayer, but there are also proteins as well, okay? And these protein molecules actually can move around the membrane. So that's why it says they can float around like icebergs. And they can be either peripheral or integral, okay? So those are two main categories of membrane proteins. We will then talk about different specific types of membrane proteins, but these are two main categories. So peripheral proteins are found on the inner membrane surface, and then integral proteins are basically partially or wholly embedded in the membrane, okay? Also, another component of the plasma membrane, and an important component, is cholesterol. Okay, so we talked a little bit about cholesterol in chapter 3 when we were talking about biomolecules. Cholesterol is involved in maintaining the fluidity of a membrane, right? So in terms of how fluid that membrane is, it sort of needs to be maintained at that sort of proper fluidity. If it's too fluid, then it would kind of fall apart, right? So it has to be held together, but you also don't want it to be too rigid. And so cholesterol is involved in maintaining that fluidity. Okay, so if we take a picture of kind of some of the components that we just mentioned, right? So here's our phospholipid bilayer. We can see the polar heads, right? Those hydrophilic heads on either side here. And then the fatty acid tails facing in towards the middle, okay? And then we can see here also in purple, here's cholesterol sort of interspersed throughout that plasma membrane. Remember, it's involved in maintaining the fluidity of the membrane. You can also see an integral protein here, which will span through the bilayer, as opposed to the peripheral proteins, which sort of hang in towards the inside of the cell, okay? They don't span through the bilayer. They're actually really associated, they're not really directly associated with the membrane. They're sort of bound to, say, this integral protein here. But again, we call them peripheral because they're sort of on the outside, they're not embedded in that membrane. Okay, so if we move on from there, there are also other components of the plasma membrane that exist. And so these are proteins and lipids that have carbohydrate chains attached to them. So we call them glycoproteins. So all a glycoprotein is a protein with an attached carbohydrate chain. Glycolipids are lipids with a carbohydrate chain attached, okay? And so these carbohydrate chains, and I'll show you a picture in a second, stick out towards the outside of the cell, okay? And they do have, you know, a sort of specific function. I'll mention that. They're sort of there as, you can think about them as sort of identifiers of the cell and involved in sort of cellular communication. So cells have to be able to identify each other. And so we can sort of think about their function that way. We will mention that again when we talk about some of those specific membrane proteins. Okay, so anyway, what I just want you to know for now is that glycoproteins and glycolipids are also part of the plasma membrane, okay? And again, the carbohydrate chains stick out towards the outside of the cell, not towards the inside, towards the outside, okay? And it says it makes the membrane asymmetrical, meaning the outside is different than the inside face, right? And so you can see that here, right? So here, this is showing you here a glycoprotein, because here's your orange protein. Say this is an integral protein that's sort of embedded in the membrane, and then it has a carbohydrate chain sticking out. Like I said, these are involved in sort of being identifiers, right, for the cell, and so that's why they're only on the outside. Likewise, you can see here, this is a carbohydrate chain that's just sticking off of a lipid here, okay? So glycolipid, glycoproteins. Also, some other things to sort of point out that I didn't point out in the last figure. This is also showing you the extracellular matrix and also the cytoskeleton. So we talked about the cytoskeleton in the last chapter. I would, if you want to kind of think about the extracellular matrix, kind of think of it as being sort of analogous to the cytoskeleton, but outside of the cell, okay? So when you think about cells as being components of tissues, you know, you need to have some structure, right? And so the extracellular matrix sort of maintains that structure in terms of keeping, you know, cells of a specific type kind of together, okay? So kind of think of it that way. And so anyway, I think I mentioned, everything else I had already mentioned on that other slide. Okay, so you can see this more closely here, this peripheral protein. See how it's bound to the integral protein, okay? It is not directly associated with that plasma membrane, okay? All right, so this is really the, this is the figure that you would want to focus on in terms of knowing the components of the plasma membrane. Okay, so in terms of a little bit more in terms of function, right? So that last slide really went through the structure, right? All of what was shown in that last picture are components of the plasma membrane. All right, so if we're going to talk about some functions of these membrane proteins, right? I sort of talked about this before. So these are all examples of different types of membrane proteins. Remember, the integral proteins and the peripheral proteins were sort of just two main categories. Here we're talking about different types of proteins. So there are channel proteins, which allow molecules to pass through the membrane, okay? via a channel. I'll show you pictures of all these things in a second. Carrier proteins. They also allow things to pass through the membrane, but they do it in a different way, okay? But very similar to a channel protein, just in terms of how molecules will pass through. Cell recognition proteins. I mentioned this before when I was talking about these glycoproteins, okay? So these act as identifiers. If you think about the body being able to recognize foreign substances, right? And also being able to recognize their cells as themselves, right? So they have these identifiers, and those are those glycoproteins. Why is this not changing? 1 second. I don't know why this isn't changing. There we go. In terms of some more proteins, we mentioned this in the beginning of the chapter, right? In our introduction, where we're talking about capsaicin, receptor proteins. Specific molecules bind to receptor proteins. So any molecule that's going to have some sort of action on the body or action on the cell has to bind to a receptor, okay? And so it says it allows the cell to respond to signals from other cells, right? So if you think about, in that sense, maybe not something that you're taking in, like a pharmaceutical drug, but it could be a hormone that's secreted from a gland. Once it gets into the bloodstream, right, it will eventually bind to a receptor on its target cell, and that's how it will initiate its action. There are also enzymatic proteins, right? Enzymes carry out metabolic reactions. Also junction proteins, which will connect adjacent cells, okay? And that's what this says here. All right, so like I said, I was going to show you a picture of every single one, and also with each picture, there's also the definition again, and also an example, okay? So I think these are super helpful, and I usually will on the exam, ask you to give me an example of something related, say, to a channel protein or something like that, right? So here, again, I said a channel protein allows a particular molecule or ion to cross through the membrane, okay? Because remember, there are many things that cannot cross through the membrane on their own, okay? Therefore, most things will cross through that membrane through channels, right? Or through a carrier protein, which we'll mention next, okay? So this is exactly as it says. It's a channel, okay? So one example of something related to an ion channel or a channel, right, is in the genetic disease cystic fibrosis, okay? It's actually caused by a faulty chloride channel. And so because the chloride ions aren't able to cross through that channel like they should, it also affects the transport of water, and that's how you end up getting this thick mucus that will collect in the respiratory pathways and also in the pancreas and in the liver ducts. And so it's a pretty serious disease. All right. Another type of membrane protein, a channel protein. So like I said, it's also going to move ions or molecules across the membrane, but instead of it being like a little tunnel or a channel, it just does it in a different way. So really the ion or the molecule will bind and then it gets released on the other side. Okay. So same idea. This mentions this is kind of I don't know, it's not the best example, I guess we could give. It says the inability of some persons to use energy for sodium potassium transport has been suggested to cause their obesity. To be honest, I'm not sure how much what the data looks like in terms of the research on this, but it's something we could look into more if we were really interested in it. But essentially, there are other examples here as well. If you just like in that cystic fibrosis example, if you had a faulty carrier protein, right, it's going to then interfere with some physiological process, okay? If these molecules or these ions cannot get across the membrane like they should, okay? So this is an example the book gave. Like I said, I'm not that familiar with the research, and I don't know how much literature there is on that, so it might be something interesting to kind of look up. All right. Cell recognition protein, right? We mentioned glycoproteins. And so this is an example of kind of cells being able to recognize, or these being as identifiers to a cell, right? So there's something called the MHC, which is the major histocompatibility complex. And so these are different for each person. So this is like you get an organ transplant, right? And they're difficult because a lot of times your body will reject that organ because the body can identify that as not being from the same body, right, as being foreign. And so these glycoproteins, again, are necessary as identifiers, okay? So it says cells with foreign MHC glycoproteins are attacked by white blood cells, right? And so that's responsible for immunity. So usually that's good. If you're talking about an organ transplant, it makes things very difficult, okay? All right, receptor protein. We mentioned this as well. And so you can see here, you have your molecule. And if you want to go with the example that we gave in the beginning of the chapter, this is capsaicin, binds to its receptor sitting on, a pain cell in the mouth. And essentially, once it binds, it exhibits some action of the cell and basically can lead to causing, pain. Here, the example we give, it says pygmies are short not because they do not produce enough growth hormone, right? But because their hormone receptors are faulty. So say this was growth hormone, it's supposed to bind to this receptor, but if this receptor is faulty and it's not able to bind, then growth hormone cannot have its action. Therefore, there will be a deficiency in growth, okay? So super important that obviously receptors function properly. Enzymatic protein, okay? We're going to talk more about enzymes in the next chapter and in subsequent chapters, but we mentioned them before. This is to give you an example of a membrane protein called adenylate cyclase, and it's involved in ATP metabolism, okay? And so cholera, which is basically a bacteria, right, that can cause disease, releases a toxin that will interfere with proper functioning of that enzyme. Okay, and so what happens is that sodium and water leave the intestinal cells and basically causes severe diarrhea, and you can actually die from that. All right, here's an example of tight junctions, or these are examples of proteins that are involved in, they're junction proteins, right? So involved in keeping cells together. So again, if you think of cells of a particular tissue, like epithelial tissue that will line your intestinal tract or something like that, it's important that these cells are very close together. And so there are specific proteins that join these cells, okay? So this gives an example. It says, as when a tissue pinches off the neural tube during development, right? So again, this is kind of giving you an idea during development that you have to have these sort of cells working together and held together to start developing organs, right? And developing the nervous system, okay? So it's super important that these junctions are maintained. All right, so anyway, these are sort of lengthy examples, so I apologize for that. If on the exam, I said, define what a channel protein is, give me an example of why channel proteins are important, right? And so it kind of leaves it sort of open-ended for you to kind of come up with your own example or just to explain why it's important in terms of what it does, okay? So anyway, those are some examples there. Okay, so now we have to start to kind of talk about what can get through that plasma membrane and what can't, right? So we call the plasma membrane selectively permeable. And by the way, a lot of this chapter, especially what we're going to talk about kind of in the next probably 10 or so slides, really pertains to that diffusion and osmosis lab, right, for this set of modules as well. So when we say it's selectively permeable, again, that basically implies that some substances can move across and so others cannot, okay? So in general, small, non-charged molecules, right? Things like carbon dioxide, oxygen, glycerol, alcohol, they all freely cross the membrane, okay? They can pass through the phospholipid bilayer and freely cross through, okay? And so it says they'll follow their concentration gradient, meaning they will move from an area of high concentration to low concentration, right? We call that diffusion. And again, they're able to just move through that membrane on their own. They don't need a channel protein or a carrier protein, okay? Water, okay, actually moves across that plasma membrane through special proteins called aquaporins, okay? Water technically could diffuse across that plasma membrane, but it wouldn't be able to do it at a fast enough or a high enough rate in order for cells to maintain function. And so water moves through the membrane through aquaporins, okay? You should remember that, okay? So it says to speed up water transport. Ions, polar molecules, okay? Usually is assisted by carrier proteins or ion, right? Or channel proteins. I already gave you those examples as we were talking about that before. It says here, some molecules must move against their concentration gradient, right? And that's called active transport. We'll talk a little bit more about that. So there are definitely examples of this where basically the cell needs to move ions against their concentration gradient, okay? And we call that, again, that then requires energy, and that's why we call it active transport, okay? The cell also has the ability to bring in or release large particles, okay? And so we'll call, can kind of generally call this bulk transport. But if we want to kind of take two sort of general terms or more scientific terms, I guess, exocytosis is basically the transport of a large particle out, and then endocytosis is basically that cell taking in a large particle, okay? So these are things that can't just move through a channel or anything like that. They're much larger. Okay, so this is basically just a figure showing you what we just said on that last slide, sorry, right? We can see here's your plasma membrane. We can see that charged molecules and ions, they cannot cross through that plasma membrane on their own. They must move through channel proteins, okay? Water, it moves through the membrane through a specialized channel called an aquaporin. Non-charged molecules that are small can cross, right? We gave examples like oxygen, carbon dioxide. They diffuse across the membrane on their own. Large molecules like a macromolecule has to move in via exocytosis or endocytosis. That's what we talked about before, okay? That you could see this is way too big to be able to move through a channel. Even if you think about like a bacteria or something like that a white blood cell needs to break down, right? Remember when in the last chapter when I was talking about lysosomes, okay? The white blood cell would engulf that bacteria, bring it into the cell via endocytosis, and then it would fuse with a lysosome so the lysosome could break it down, okay? So that's that type of transport. Okay, so this is a table that basically sort of summarizes what we just said and some of the things that we're going to talk about a little bit more, right? So diffusion here, okay, the direction it moves is toward lower concentration. When we talk about diffusion, it's molecules moving from high concentration to low, right? Or moving along their concentration gradient, okay? That's what this says here. And so this is just giving examples of those things that can just diffuse across the membrane on their own. If you just think about diffusion in general, right, and there's no membrane there, it's the movement of molecules along their concentration gradient. Here we're particularly talking about moving across the cell, right, diffusing across that plasma membrane, right, into and out of the cell. Facilitated transport, again, things are moving along their concentration gradient, except that they have to move through that membrane through channel or through a carrier protein, okay? Active transport, this is now going in the opposite direction, right? It's moving against the concentration gradient and it requires some sort of carrier protein, but it also requires energy, okay? So things like sugars, amino acids, and certain ions will move this way as well. Bulk transport is, so it gives you an example of macromolecules. I also gave you the example of like bacteria or something like that. And this can move either way, right? And it says it utilizes vesicles. If you think about that example that I gave with the white blood cells and the bacteria and engulfing it, remember that involves vesicles. So if you're kind of not remembering what I'm talking about, if you go back to chapter 4, when we were talking about the lysosomes, that's when we talked about that a little bit more. So it might just help you to kind of have that example. Okay, so now let's talk more about the process of diffusion, right? And passive transport. So the first part of this just talks about diffusion in general, right? Which I kind of already mentioned. And then we talk a little bit more about moving across the membrane. Again, we've sort of already talked about it, so let's clarify it a little bit more. So if we want to talk about a solution, right? A solution contains some liquid, usually water, right? A solvent, and a solute. Some dissolved solid, maybe like sodium chloride, right? A salt. If we're going to talk about the general term of diffusion, it is the net movement of molecules down a concentration gradient, along their concentration gradient, from high concentration to low concentration, however you want to say it, okay? But always remember that the molecules are moving both ways, but we talk about net movement, okay? Now, as these molecules move along their concentration gradient, eventually equilibrium will be met, and that is when the net movement stops. So again, there is still movement sort of back and forth, but the net movement has stopped. Okay? So it says solute concentration is uniform. There is no longer any concentration gradient when equilibrium is met. Okay, so this is like a super simple, right, illustration here. Here's your fish tank. you put a little dye molecule in that fish tank, and everyone always asks, or wonders if the poor fish are okay when you put this dye molecule in there. But anyway, you can see it starts off here, right? This dye molecule placed in the water, and what's going to happen over time, right? These molecules of dye are going to start to diffuse out into the water, right? Because again, it's high concentration to low, okay? And eventually equilibrium will be met. You have equal distribution of these molecules, okay? So there you go. That's just basic diffusion. Now, again, remember, well, we're going to talk about that yet. So again, what we're really focusing on here in biology, we're not going to talk about general kind of diffusion. We're going to more focus on diffusion across the membrane, right? And what happens when we're talking about molecules moving across the membrane, all right? So here's an example. I mentioned to you that oxygen and carbon dioxide can diffuse across the membrane on their own, right? So if you think about this kind of bigger picture here, this is the alveoli of the lungs, right? And so you got lots of oxygen in there when you're inhaling, right? And so what happens just based on the concentration gradient here, there's a high concentration of oxygen in the lung, right? And a low concentration in the blood here. Oxygen will move along its concentration gradient purely by diffusion and end up in the blood vessels, right? In the red blood cells, okay? And again, it just moves by diffusion across the membrane because it can diffuse readily across the membrane. So, sort of a simple concept for a complex sort of process, I guess, or a seemingly complex process, I guess. Okay. So, like I said, we're going to mostly be talking about movement across a membrane. If we're talking about biology and we're talking about living things, we're talking about cells, right? So what's osmosis, right? It says it's a special case of diffusion. Osmosis is the diffusion of water, okay? So we're not talking about the solute moving. We're talking about which way water moves, okay? Because remember that membranes are selectively permeable. And again, this is illustrated in lab. That experiment where we have this dialysis tubing that simulates a cell and simulates a selectively permeable membrane, okay? So again, because certain things can't move through a membrane, right? But water can, water will move to basically, try to reach equilibrium, just like we sort of talked about in general with diffusion, okay? So again, when you're thinking about osmosis, you're thinking about diffusion of water, you're thinking about which way water moves, okay? And you're thinking about the concentration of water kind of outside of the cell versus inside of the cell, right? What that concentration is. And so, so again, I said all this, okay? So it says here, solute concentration on one side is high, but water concentration is low. So if you have solute concentration being high, that means water concentration is low. If on the other hand, solute concentration is low, but water concentration is high. Okay, and so water, just like any other molecule, will move from high concentration to low concentration. Okay, so again, when you're thinking about osmosis or talking about osmosis, you're following the water. You're not following the solute. Okay, so that's what I said this already too. Water can diffuse both ways across the membrane, but the solute can't. Okay, so again, the net movement of water is towards low water concentration. Okay, because again, it's moving from high concentration to low. If you then want to think about the solute, so where there's low water, there's a high amount of solute. Okay, so they still relate to one another, but again, osmosis, we're talking about water moving. Okay, and this is something that you have to think about a lot in biology, again, because we talked about, the fact that some solutes can't get across the membrane, water can. Okay, all right. So something called osmotic pressure, if you hear the term osmotic pressure, it's the pressure that develops due to osmosis, meaning the movement of water or the diffusion of water. Okay, so this is actually something that sometimes you can do as a demonstration in lab. But basically, you have, so here, right? In the flask here, you have 10%, a 10% concentrated solution, okay? So it says there's a higher percent of solute here, meaning there's a higher amount of solute and there's a lower amount of water, okay? Here on the outside, right, it's only a 5% solute concentration, okay? So there's more water here, right, and less solute. So now if you just follow the movement of water, water is going to move from, sorry, my mouse froze. Oops. Water is going to move from high concentration to low, right? So water will move from the beaker into this flask here, okay? If it's following its concentration gradient, right? And so that's what this is showing you here, okay? You basically end up with more water in here because it's following its concentration gradient, okay? And again, the solute, because this is a selectively permeable membrane here, only water can cross, not the solute. So if it's trying to reach equilibrium, water will move until that equilibrium is reached. Okay, now there are some terms for these solutions, okay, in terms of how much solute is there versus how much water, okay? And again, this was addressed in the lab as well, okay? So we will call an isotonic solution the case where the solute and the water concentrations are equal on both sides of the membrane, okay? So it's the same concentration of solute and the same concentration of water inside the cell versus outside the cell. And so in that case, there is no net gain or loss of water, right? Because there is no concentration gradient. All right, so that's isotonic. Hypotonic, the concentration of solute in the solution is lower than inside the cell. So outside of the cell, okay, it's less concentrated, right? Meaning there is more water outside of the cell than there is inside of the cell. Okay, so it says the concentration of solute outside of the cell is lower than inside of the cell. So if the concentration of solute is lower, then the concentration of water is higher. Okay, so you have to just always think about both. So in this case, it causes something called turgor pressure. And again, I addressed this in lab because water is going to be drawn into the cell, right? So in a plant cells like this because they have the cell wall to protect them. Water is drawn into the cell. It fills up that central vacuole and helps maintain turgor pressure. However, in animal cells, it can actually cause those cells to lyse or rupture, okay? Now, on the flip side of that, if we talk about a hypertonic solution, okay, the concentration of solute is higher outside than inside. So hyper concentration of solute is higher, okay? However, if the concentration of solute is higher, the concentration of water is lower, okay? So now in this case, the concentration of water outside the cell is lower, inside the cell is higher. So if water moves from higher concentration to low, it's gonna move from inside the cell to outside the cell. And if water is drawn out of a cell, it will basically cause those cells to shrink, okay? And we call that crenation in animal cells. And again, I brought this up in lab, and plasmolysis in plant cells, okay? Not a good scene. All right, and so this picture addresses or illustrates basically everything I just talked to you about, okay? So in an isotonic solution, no net movement of water, animal cells, super happy. Same thing, there's no net movement of water in plant cells. In a slightly hypotonic solution in a plant cell, that's good for them. It helps them maintain turgor pressure, and an animal cell can cause that cell to lyse, okay? Hypertonic will cause water, right? Water's gonna be leaving the cell. Okay, more water is gonna leave the cell than coming in, causes the cell to shrivel, okay? All right, now, so you may need to take a little bit of time to really think about those concepts I just talked to you about, okay? Super important to understand osmosis and understanding what an isotonic, a hypotonic, and a hypertonic solution is, and which way water is going to move, right? And then what effect that is going to have on the cell, okay? Super, super important concept. So if you need me to go over that again, I can, but you may have to maybe watch, there may be some animations on Moodle on that. You probably can search up some other videos on that if you need a little extra on that, okay? But I'll tell you that it's a super important concept. All right. So that was osmosis, okay? That was the movement of water across the membrane. That's what we just talked about. Now, we mentioned this in that table, right, a few slides ago. So facilitated transport, right? This is molecules that cannot cross the membrane on their own. They have to go through a channel protein or a carrier protein, okay? They're still moving along their concentration gradient though, okay? So they're moving from high concentration to low. However, unlike carbon dioxide and oxygen, they cannot diffuse on their own. They must move through a carrier protein or a channel protein, okay? So that's all facilitated transport is. Again, the molecule still moves along its concentration gradient. It just does it with the help of a channel protein or a carrier protein, okay? So that's what this is showing you here. Here's your solute outside, right? High concentration outside, sorry, let's use my mouse. High concentration outside, low concentration inside. These molecules have the tendency to move this way. They cannot cross that plasma membrane on their own. They have to move through a carrier protein. So we call this facilitated transport, okay? Again, however, they're still moving along their concentration gradient. It does not require energy. So it is still a form of passive transport, okay? All right, so this is going to show you it moving through, that molecule moving through. All right. Now, I just told you that facilitated transport, though it involves a carrier protein or channel protein, it is still passive because it does not require energy. Active transport, on the other hand, is transport that requires energy. And molecules move against their concentration gradient, right? So if you need to move something in a direction that it does not want to go, it's going to require energy to put in, right? I always give the example of like, I'm walking my dog, right? But he doesn't want to go. I really have to pull him to make him come in this direction if he's trying to go the other way, okay? As opposed to, you know, again, something moving along in that same direction, relatively speaking, doesn't require much energy, okay? So again, active transport, you're moving molecules against their concentration gradients. So in this case now, you're moving from low concentration to high, okay? It is also facilitated by carrier proteins, but again, because they're moving against their concentration gradient, it requires ATP or energy and is thus active transport. Big example of this is the sodium-potassium pump, okay? We call it a pump because it requires energy, okay? It uses ATP to move sodium ions against their concentration gradient and potassium ions against their concentration gradient. Why does it do this? Because it maintains a certain charge across the membrane that is super, super important to the cell, okay? And, you know, it's not something that I get into in great detail, but the sodium-potassium pump, if it does not operate, basically your cell can't function, okay? All right, so this is what it's doing. Again, why is it important? Because it maintains the charge across the membrane, okay? All cells have a certain charge across their membrane. And one of the things that's important in maintaining it is the sodium-potassium pump, okay? So, I mean, you follow this along, it's going to show you how it operates, right? So it's showing you here, you got your 3 sodium ions that are binding, right, to this pump, this carrier protein. And you can see here you got ATP. This requires ATP. Those 3 sodium ions from inside the cell that are bound are going to move to the outside of the cell. Now, again, they're moving against their concentration gradient here. And then on the flip side, you have 2 potassium ions that will bind here, and they will move from outside of the cell to inside of the cell. Okay? So it's moving 3 sodium ions out of the cell and 2 potassium ions into the cell. All right. Now, I mentioned this, right? This bulk transport, right? And the 2 main ways that the cell, or 2 main ways of, or means of transport, I guess, of large particles that cannot go through these carrier proteins. Exocytosis, meaning it's the cell secreting contents. So this is the example of, if you think about back to chapter 4, when we were talking about the endomembrane system, right? And cells that are made on ribosomes in the ER that then move to the Golgi, right? And then they get packaged into a vesicle and are secreted out of the cell. That's exocytosis, okay? Endocytosis, on the other hand, is taking in a large particle, right? So if you're going to talk about a white blood cell taking in a bacteria, it's endocytosis. there are two specific types of endocytosis, phagocytosis and pinocytosis. So the example of a bacteria being taken in, that's an example of phagocytosis. Pinocytosis, sometimes people call it cell drinking. So it's kind of taking a bunch of small particles in at once, okay? And then there is also receptor-mediated endocytosis. So this is specific, meaning something will bind to the receptor and then it will be taken in, okay? So just a more specific type. All right, so this is showing you, let's look at exocytosis first. So again, think about these as proteins, right? That were just made by the cell. They're secreted proteins. That vesicle will fuse with the plasma membrane and then they'll be released to the outside, okay? Exocytosis. Endocytosis, remember there are three different types of endocytosis. Phagocytosis, where it's taking one big particle in, say like a bacteria, that needs to be broken down. You can see this this is showing you so this is probably an amoeba. So these are two single-celled organisms. Let's think about them. But they're eukaryotes, but anyway, they're single-celled organisms. So this amoeba will basically take in food via phagocytosis. And so it's basically going to engulf this paramecium, which is another small eukaryote. Okay? Here, this is showing you pinocytosis, right? It's just taking in a whole bunch of little particles. Okay? I told you sometimes you call it cell drinking. Okay? And then here's receptor-mediated endocytosis. This is more specific, meaning these molecules have to bind to the receptor, and so they have to match that receptor before they're then taken in, okay? And these are all, electron micrographs showing you these processes. Okay, all right. Finally, the last part of this chapter is talking about modifications of cell surfaces. So talking a little bit more, actually mentioning the extracellular matrix, right? I kind of mentioned that because when you see those plasma membrane pictures, you see the extracellular matrix on the outside and then the cytoskeleton on the inside. And we really didn't talk about the extracellular matrix yet, okay? So it says here in terms of a definition of the extracellular matrix, meshwork of proteins, right, and polysaccharides, right, carbohydrates that are in close connection with the cell that produce them. So things like collagen, okay, elastin, okay, integrin, these are all types of proteins that exist in the extracellular matrix. So if you think about tissues like, or connective tissues, things like bone, things like tendons, cartilage, okay, they have these types of proteins exist in the extracellular matrix, and it's a super sort of strong, structured extracellular matrix that then the living cells kind of sit in, okay? So that's my sort of example there. So if you look at some of these, collagen resists stretching, elastin provides resilience, okay? Integrin, actually, these integrins actually play a role in cell signaling, okay? But these all exist outside of the cell, okay? Proteoglycans will help regulate the passage of material through the extracellular matrix, then to the plasma membrane, okay? So the extracellular matrix, I guess you can kind of think of it as an extension of the cell, okay? It's outside of the cell, it's not part of the cell, but it's closely associated with it. I guess that's the best way to think about it, okay? So, and this is showing you this here. So again, here's your cytoskeleton, right? We talked about that, right? Being composed, remember, of actin filaments, intermediate filaments, and microtubules, right? And the actin filaments are the ones that are really close to the plasma membrane. Okay, so you can see that these integral proteins are actually bind to the cytoskeleton. And if you look, they're actually also then bound to the extracellular matrix, some of these proteins that I mentioned in the last slide, okay? So this is going to anchor that cell, right? And link that cell to sort of the outside, okay? So it's not just that the outside of the cell is just like, I don't know, like air or some liquid. There are structural and proteins and components there that then link the plasma membrane to it. Okay, so again, when you're talking about the plasma membrane, you have to talk about the extracellular matrix because they are closely associated, and you can see that in this figure here. Okay, so just a little bit more in terms of joining cells together, okay? And so these are just literally, that's it. There's a couple more slides here. These are different types of junctions between cells, right? So, and I don't know that, I mean, I wouldn't like spend tons and tons of time studying the sort of subtle differences between these different types of junctions. But let's just kind of take it as sort of big picture in terms of, so adhesion junctions, okay? These I'm giving you two different examples of adhesion junctions. I think the one to think about is tight junctions. So think about adhesion junctions or think about tight junctions as junctions between cells that basically form impermeable barriers. So things cannot get through. You do not want things to get through these junctions, okay? On the other hand, there are junctions called gap junctions. And these are really basically channels within the plasma membrane that allow things to directly pass from cell to cell, okay? So that's what this is telling you here. So if you look at this picture here, so like I said, I kind of, you can kind of, the adhesion junction, you can see, is showing you different sort of proteins that will join together here. And you're going to have different types of adhesion junctions. Again, one type of those is a tight junction. And again, in a tight junction, there is not a lot of space here, as you can tell, right? These two, so think of this is one cell, plasma membrane of one cell, and this is a plasma membrane of another cell. They are squeezed super, super tightly together so that nothing can cross through, okay? So that's the idea, and that's sort of what I want you to think about. I'd rather you sort of think about tight junctions versus gap junctions, okay? Because there are other types of junctions as well. There are other types of proteins that join cells together, and you can get really complicated here. So let's not get complicated. If you look at this here, I mean, I hope we can see the difference between a gap junction and a tight junction, okay? There's no channel that's going to allow things to directly, pass from this cell to this cell. However, in gap junctions, that's exactly what happens. Ions can basically flow freely from, this cell to this cell. And this is super important, actually, when you think about cardiac cells and heart muscle cells, in terms of allowing for that coordinated contraction that's necessary for the function of the heart. It's because those cells have gap junctions that allow ions to flow through, and that basically causes these muscle cells to contract at the same time or in a coordinated fashion. Okay, tight junctions, I like to give the example of epithelial type tissue, like tissue, your skin, right? Or that epithelial tissue that lines your intestinal tract, super tightly packed together. Okay. Also worth mentioning, because for plant cells, right, they have a cell wall. And so if we talk about junctions between these cells, it's going to be a little different because the cell wall is there. Okay. So there's actually something called plasmodesmata, which will penetrate the cell wall, okay? And then that will allow passage between cells, okay? So again, you have this sort of extra complication because of the cell wall. So there needs to be these sort of gaps in the cell wall to allow for passage between, okay? So that's what this is showing you. So again, if you look at this electron micrograph, you can see here's cell 1, here's cell 2. These are the plasmodesmata that are basically spaces between that cell wall there, okay, that allow that passage through. All right, that's all. All right, guys, and that ends that lecture on chapter 5. So remember, a lot of these concepts are also addressed again in lab. I will again reiterate that understanding osmosis and understanding the movement of water across the plasma membrane is super important, okay? So there are a lot of important concepts in this chapter. Remember that you need to have your exam 2 review sheet. I should have been reminding you this throughout, but your exam 2 review sheet, right? And should be completing that with notes. So chapter 5 is the first chapter on the second exam. Exam 1 is chapters 1 through 4, and exam 2 starts with chapter 5. Thanks, everyone.